Thank you for inviting me to your webinar and also for the very kind introduction. Yeah, as you can see in my title, I will talk about bioprinting of organ models, and we intend to use these organ models for the replacement of animal experiments. All the research I will present today was, of course, not done by myself, but rather by my co-workers. And as you can imagine, this is still a picture from the Corona times. We hope to have a new picture soon, but I like this one very much because I asked my co-workers to show their creativity and to chaotic structures, and then they all aligned in a wonderful symmetric triangle, and only some foreign PhD students stepped out. So this is typical German. So what I want to go through is I will give you a brief general introduction into bioprinting technologies, and will then more specifically focus on our research. As Katrin already introduced, we do various organ models for infection studies, particularly lung models and liver models, and then we also have projects to produce cancer models. And I will have an extra section on xeno-free methods to do something we call clean bioprinting, which means to produce models for research without any animal components. Let me start with a brief introduction into bioprinting. Why do we want to do, or why do we do this very complicated, sophisticated, and expensive experiment? Well, probably everybody who's in this webinar shares the opinion that we have a translation problem with current research. So our current research paradigm says that we test things in 2D cell culture first, then we move on to animal experiments, and then we go to the clinic. And there is a long list of studies, and over several years, they all come to the same conclusion that roughly 90% of the drug candidates we test in preclinical development and that are efficient and non-toxic in preclinical animal experiments fail in clinical studies. There are various reasons for this, but among the main reasons is a lack of efficiency and unexpected toxicity. In some indications, for example, in oncology, the failure rates are even higher, reaching up to 97%. And it is actually pretty easy to understand why we have these problems. Let me just show you two examples which are relevant to the research we do later on with the bioprinting. The first is our way to conduct cancer research. So what we usually do is when we have tested something in cell culture, we go to a mouse model. And as human cancers are usually detected by the immune system of the animals, we frequently use nude mice that have no immune system. So there are two problems with this approach. First of all, the lack of the immune system. And secondly, the human tumor is embedded in an environment of a mouse. And we all know that for the cancer, the microenvironment is very important. So the mouse and microenvironment will give different signals compared to what a human microenvironment would do. Second example is the COVID research. There was a nice overview over the first 1.5 years of Corona research from the beginning of the crisis until July 21. And in this survey, the authors found that 90% of the animals that were used for Corona research were mice. And most of these mice were wild-type mice. So as we all know, the coronavirus enters human cells by attaching to the ACE2 receptor. And these wild-type mice do not even have a functional ACE2 receptor. So we adapt the viruses to our animals, or later on, that people produced transgenic animals that produce the ACE2 receptor. So then they adapt the mouse model to the human pathogenic virus. But in all cases, there is no big surprise that the causes of disease are very different when we do infection experiments in a human, or the experiments in a mouse compared to what we find in a human. That is why we pursue an alternative way of research, and that is that we produce human 3D models by bioprinting. And so I will start with a short definition. What I understand is bioprinting. Bioprinting is a specific technology from the field of 3D printing. 3D printing is a process to produce three-dimensional objects by successively adding material layer by layer. That is in contrast to what we had before, when we either had subtractive manufacturing. For example, imagine a sculptor, Michelangelo, seeing a block of marble before, in front of him, and then he sees the Pietà in there, and he removes, by subtractive manufacturing, he moves the parts that should go. The alternative is, of course, casting of three-dimensional objects. But in 3D printing, we produce something by additively adding layer by layer, and that is why the scientific term for 3D printing is additive manufacturing. And I have some colleagues that strictly avoid using the term 3D printing, but say that we really should use the term additive manufacturing, but 3D printing is, of course, the more popular term. So what is specific in 3D bioprinting? That is that we have living cells, living material during the printing process. There are also strategies to produce a scaffold and then repopulate the scaffold afterwards with cells. But in my understanding, and that is shared by most of the colleagues in the field, bioprinting really involves the living cells during the printing process, and the living cells are embedded in biocompatible material, and this material frequently also contains certain signals such as growth hormones, extracellular matrix components, and other things. So bioprinting, in this understanding, is a method for replacement, so the 1R of our famous 3Rs. Here is a typical workflow in bioprinting. So we always have to start with a computer design of our 3D model. The 3D model can either be obtained from a natural source, like to have an organ or a piece of tissue, and to do an MRI or CT of the scan of this tissue to obtain the three-dimensional structure. We can also design the models by ourselves. In all cases, we use some computer-aided design systems. And then we have two major parts for the hardware. And that is, on one hand, the bioprinter, and I will introduce different bioprinting technologies in a minute. And on the other hand, it's the bioink. The bioink is the biocompatible material that contains the living cells and that is used for the bioprinting process. So let me go through these steps. So the first part, as I said, is the cut design of a three-dimensional object. We all know that these cut programs are widely used to produce ship or cars or whatever you imagine. And then, as I said, we can either have a natural model. So here, for example, this is a trachea from a pig that was decellularized, and then we had a CT scan of this decellularized trachea. And here we have the computer model of the trachea, and then we did some 3D printing. In this case, it was only 3D printing and not bioprinting because there were no cells involved, and we could reproduce this trachea structure. The alternative, as I said before, is to produce artificial models. And here are two examples I will refer to later on. One is our model, which we currently use for the lung model. I call it the baby pool. And the second is a vascularized model where we have a 3D tissue and we have a vascular structure that is embedded into the 3D tissue, and we have an inlet and an outlet. So we can fuse our tissue model. The second step, as I said, is to create a bioink, and actually that's the big magic, the major part for a bioprinting group, to optimize the bioink. So it's pretty easy to design a nice model, and there are nice bioprinters commercially available, but the real magic thing, the real thing to work on is to produce good bioink. As I said, the bioink contains the living cells, and it's a soft biocompatible material. And this material must have two properties. First of all, it must have a low initial viscosity during the printing process, so we can start the printing. But in the second we do the printing, it must immediately become stiff to maintain the structure that we intend to print. There are different types of bioinks. Most commonly, hydrogels are used as bioinks. So hydrogels are polymers that retain large quantities of water, and they can be cross-linked. Typical bioinks used in bioprinting are agarose and, for example, gelatin. I will show you our usage of some of these bioinks with these components, and alginate is another natural source for a hydrogel. In addition, there are some synthetic hydrogels like PLA, PLGA, or polycarprolactone. The synthetic hydrogels have the advantage that you can modify their characteristics by chemical synthesis. However, in many cases, the biocompatibility of natural hydrogels is better than that of synthetic hydrogels. Here are, as I said, two typical examples. The alginate, it's a polysaccharide, and gelatin, which is the denatured form of collagen, so what is present in the extracellular matrix. And many studies use mixtures of alginate and gelatin for the bioink. Okay. Then the second important part is the bioprinting technology. And while we have several dozens of technologies out there for conventional additive manufacturing, there are 4 major technologies for bioprinting. The most commonly used technology is the extrusion or microextrusion bioprinting. Then we have the inkjetting, laser-induced forward, and stereolithography. So microextrusion printing means that we have our bioink in a cartridge, and then we put on some pressure. In most cases, we use pneumatic extrusion, but we also sometimes use a mechanic microextrusion, and extrude the material through a thin nozzle. Inkjetting means that little droplets of our biomaterial are released from the cartridge by either in thermal or a piezoelectric actuator. Then we have the laser-based technologies. One is the laser-induced forward transfer. In this case, the bioink with the cells is placed on a ribbon, and specific positions of the ribbon are then addressed with a laser beam, and a droplet with the cells is released from this specific position. This technology is not so commonly used as the other 3 I have listed here. So the other 3. So the last technology I want to introduce is the stereolithography. Stereolithography means that we have a liquid material that is polymerized by a laser beam that addresses a certain position to be solidified to produce the 3-dimensional object. A specific variant of stereolithography is digital light processing, and I want to mention this here because later on I will show you a study where we use digital light processing. In this case, we don't only use a thin laser beam that solidifies material at a certain position, but we solidify the whole layer at once with a mirror technology. So there are 2 important things to take home from this part of my presentation. The first is there are various technologies for bioprinting out there, and none of those is the very best technology, but rather each technology has its advantages and disadvantages. For example, the DLP printing has a very, very high resolution, much higher than, for example, extrusion printing. But on the other hand, you need an initiator for the radical reaction, and that might be toxic to the cells. Plus, you always irradiate your cells, and in some setups, this irradiation has a high energy and may be harmful to your DNA. So it always depends on what you really want. Do you want to be very cell-friendly? Do you want to have a high resolution? Do you want to include multiple materials? Do you want to have a single material only? So each of these applications needs a different bioprinter. The second important thing to take home from this part is that the bio ink always has to be adjusted to the technology you use. For example, in microextrusion bioprinting, we commonly use these alginate and gelatin mixtures. For the DLP and stereolithography printing, we use methacrylated gelatin, GELMA, which is commonly used for these technologies. So technology and bio ink always has to fit together well. So to be not to be too theoretical, I brought two little movies of our bioprinter. One is the Cell Ink BioX printer, and it works. So here we have the cartridge with our bio ink in the cells, and we have a pneumatic extrusion. So there is air pressure on this cartridge, and then we produce our three-dimensional model. What you can see here is that we have two cartridges with two different materials, and obviously there are more positions available. So this, for example, just printed a little cancer, and now comes the second cartridge that produces healthy material, healthy tissue surrounding the cancer. Another big advantage that is already can already be seen here is that we can produce a large set of organ models with high precision and high reproducibility. So here, for example, we have it's a 24-well plate, so we can produce 24 cancer models that are almost completely identical, which is of course different to animal models which differ from animal to animal. This one is a different extrusion printer. The principle is pretty similar. What you can see here is that we have a very thin and fine nozzle, so we can print with very high precision. However, the disadvantage is with the thin nozzle, you always have a higher shear stress to the cell. So again, here you have to optimize something. Do you want to improve your resolution, or do you want to minimize cellular stress during the printing process? Here is an example of a stereolithography printer. As I said, you have liquid material, and then comes the laser beam, solidifies the material for one layer, then it's washed away. Next layer is produced. Again, here you see the laser beam. The next layer is produced and it's washed away. And this process is repeated over and over again. Okay, as I said, there are different technologies out there, and as a consequence, we do have a set of different printers. Here, for example, you see a digital light processing printer. These are 3 generations of the cell ink extrusion printers. This is a very sophisticated high-end printer which can accommodate different types of technologies like pneumatic microextrusion, mechanic microextrusion, and also inkjetting. Here you have 2 stereolithography printers. This one is a simple consumer printer, which we basically use for lab courses. And this is my favorite printer because this one prints chocolate. And so here are some of the 3-dimensional objects we produce. Here you see this nice chocolate horse that we printed. This is an example from our lab course where we took pictures of one of the students, and then the students learned how to arrange these pictures to give a 3-dimensional object that was then printed with this FDM printer. Here, these are the more biologically relevant models. I've already shown you the trachea here, and here you see an example of a vascularized tissue model. I will come back to this later in my presentation. Let me now come to our results, and I will first start with the lung model. So the funny thing is that when people speak about 3D bioprinting, 90% of the studies use a simple grid model. And so did we in the beginning. And this grid model has the advantage that you already produce your cells in a three-dimensional arrangement, and you have all these holes so that all the cells are in close contact to the media, and nutrition and oxygen supply is very good. Still, as I said, one of the major tasks in bioprinting is the optimization of the bioink. And here we use a basic composition of alginate and gelatin, and we added different concentrations of matrigel to this bioink. And here you see, for example, if we do not add any matrigel, after 7 days, all cells sedimented to the bottom. Whereas at 20% matrigel, you have a nice three-dimensional distribution of the cells. And we also did some SEM images. And here you see that we have, with the intermediate matrigel concentration, we have a nice porosity, and that means good supply of nutrients for the cells. Of course, the major readout, more or less, is the cell viability. As I said, the cells experience shear stress during the printing process. And so we did a live dead stain. The living cells are stained in green. The dead cells are stained in red. And here, again, you see that at intermediate concentrations, you have a good cell viability. At too high concentrations, you have a lot of red dead cells in your model. And this is also reflected here in the metabolic assay. It's an XCT assay where we measure the metabolic activity of our cells. And let's go to day 7. Here we have, again, the 20% matrigel. We have the highest metabolic activity of the cells. Our background actually is that we are infection biologists. So we have a long-term experience with virus research. But as I mentioned in my introduction, we think that the animal models do not reflect well the cause of disease in a human infection. That's why that is our motivation to produce these bioprinted organ models. And so we infected our organ models with influenza virus. And here you see the results from a 2D cell culture. And in our 3D models, the virus also replicated nicely. And at least to our research of the literature, that was the first experiment carried out to do infection studies in a bioprinted organ model. Another interesting aspect was that when we stained the virus proteins, we found the expected even distribution of the infection in 2D culture. But in our 3D model, we find clusters of infection. So some cells become infected and the infection spreads around from these infected cells. And this is more likely to represent the situation in a human lung than the two-dimensional cell culture, which has an even distribution of the infection. Okay, we then developed our lung model further. In the first generation lung model, we only had one cell type, a cell line representing the alveolar epithelial cells. And in the second generation of our lung model, we used 3 different cell types. So we had primary human lung fibroblasts that we mixed with THP1 cells. So this is a monocyte cell line. So this represents at least some type of an immune system. So we had 2 basic layers of fibroblasts and the immune cells, and then we had the top layer with our epithelial endothelial cells. So there's multiple cell type model. And so one interesting observation was that the different cell types improve cell viability. So here we have the time course of our 3 cell type model, including the A549 fibroblast cells and the THP1 cells. And we cultured the model over 35 days. So here we already have a big advantage of the three-dimensional models, which we can culture for a very long time. 2D cultures are usually only good for a few days, and then you can't use them anymore, whereas the three-dimensional culture can be used for more than a month. And we have a very high, even slightly increasing cell viability in our multi-cell type model. In contrast, if we have the A549 cells only or the primary fibroblasts only, we have a steady state or even a decrease in cell viability. So there is a positive effect on each other in the multi-cell type model. Another thing is that we observed some nice morphological things. For example, here you see that in the 3-cell type model, we have a clustering of the A549 cells on top of this model, whereas in the A549 only model, the cells remain spread and distributed. So obviously there is some interaction between the different cell types in the 3D model. And then again, as I said, we are interested in infection biology. So we used our influenza A viruses and we tested infection. So here we have models without alveolar epithelial cells, and we don't observe any infection. And only in the models that have a top layer of the epithelial cells, we find infection of these epithelial cells. We then tested a virus inhibitor, oseltamivir, so that's the common influenza inhibitor, and we found a nice dose-dependent decrease in virus titer by treating with increasing concentrations of the antiviral drug. And we also tested for the expression of interleukin 29 and found that our model produces a rather high level of the interleukin. So there is some type of an immune reaction. And when using the antiviral treatment, we can also suppress not only the virus, but of course also the immune response to the virus. Now we move on to the next generation of lung models. In this case, there are two important points. First of all, we have a fourth layer of endothelial cells. So this is like in the natural lung, where you have on one side the endothelial cells, then you have the fibroblasts, and then you have epithelial layer. And the second important thing is that we culture these models as at an air liquid interface in Ali cultivation. So this is of course also what we have in the biological lung, where the lung has contact to the air on one side and to the blood on the other side. So here we have this Ali cultivation. Actually, for the experiments, we then use the baby pool, which I've shown you before, because this nicely retains the top layer of epithelial cells. So we printed our model. We then seeded the epithelial cells on top in this baby pool, and then transferred it to the air liquid interface, and then carried out our experiments. So here you see that we have our three-dimensional construct with the top layer of the epithelial cells. Here it's A549 cells. And the here on the right, you see the DAPI stain of the nuclei. So first of all, one nice observation was that when we culture our models at an air-liquid interface setup, we observe production of mucus, which is of course good because that is also what happens in the lung. And it is hardly visible, but you can see it in the original figure. We also see production of pan-CK as a marker for the epithelial cell. Initially, we used A549 cells, but since we now move into corona research, these A549 cells are no longer useful as they do not produce the ACE2 receptor. And we then switched to CALO3 cells. So the first nice thing was that they arranged into a very, very thin layer, which also already comes close to the monolayer we have in the biological lung with the epithelial cells. We also see some mucus. Of course, it's less because we have less cells, but it exists. And then we have the ACE2 receptor. And now we already carried out some research in collaboration with a group of Christian Drosten, and they infected our model with coronavirus. And we have some first preliminary results. Unfortunately, they're not ready to be presented, but in the initial results indicate that the coronavirus nicely replicates in the CALO3 cells here in the top layer of our model. Okay, let me now switch to our liver model. And I will here already briefly introduce our efforts to replace animal components in non-animal methodologies. I will come back to this later on again. In our initial lung model, which I showed you, we used matrigel. And everybody who does stem cell research, most of the people who do three-dimensional organ models or organoid research, and also those who do bioprinting, everybody uses matrigel and then claims, oh, I do a replacement experiment for animal experiments. But as some of you may know, matrigel is obtained from a sarcoma that was implanted into a mouse, and then the sarcoma is harvested, and the matrigel is the extracellular matrix that can be obtained from this sarcoma. So to replace the use of matrigel for our next study, the liver model, we used an extracellular matrix from a human donor. And then we used the bioink for the liver model with HEPA or G cells, which are the best cells, immortalized cells. So it's the best cell line for liver experiments. This probably looks pretty familiar to you. We first optimized our bioink again. We optimized the addition of human extracellular matrix, the three-dimensional distribution of the cells, the cell viability, and the metabolic activity, and then had our optimal conditions, which contain 0.5 to 1 milligram of human ECM in the bioink. For the liver, the nice thing is that you can measure different markers. For example, we measure albumin secretion or cytochrome P450 3A4 expression, and we find our models to express these markers at high levels. And then again, we also did some infection and also some transduction studies. As you may know, there are intensive efforts to use viral vectors for gene transfer in gene therapeutic applications. And the most promising vector here is an adeno-associated virus. So we had an adeno-associated virus that produces GFP as a reporter gene, and we transduced our liver model with the AAV vectors and found a nice and high expression level of the reporter gene. Our AAV vectors also produced a short hairpin RNA, and that mediated 80% inhibition of our of the targeted endogenous gene, which was human cyclophilin B in our liver model. So these models can be used to optimize viral vector transfer of transgenes or silencing approaches in a three-dimensional setting. In addition, we also carried out infection studies. Here we used human adenovirus 5. This is commonly actually associated with conjunctivitis or a mild cold, but for immunocompromised patients, human adenovirus 5 may become very dangerous. Without immune system, it may severely damage the liver, and that might have a lethal outcome. So we carried out some infection studies in our bioprinted liver model, and we found that the adenovirus nicely replicates in the liver model, so we can use the liver model to further develop antiviral strategies for human adenovirus. Here in these experiments I've shown you here, we also used this grid structure, but then we improved the structure. And as I said in the introduction, each technology has its advantages and disadvantages. And here we use the digital light processing bioprinter, and we use methacrylated gelatin as a bioink. And here you can see the natural structure of a hepatic lobule. It's a hexagonal structure. And with this high-resolution bioprinter, we can reproduce these hexagons. Here is a microscopic view and a macroscopic view. We can reproduce these hepatic structures at high resolution. What we currently do is that we include different cell types into our liver model as well. Let me now switch to our cancer model. As I mentioned in the introduction, cancer is a topic with a particularly low translatability between preclinical research and clinical trials. I mentioned the number of approximately 97% of all candidates that fail in oncology. And this is, for example, in parts at least, due to the artificial murine microenvironment in which human tumors are tested. So what we do is that we print a human tumor that is surrounded by human cells as the healthy tissue. What we started with was a neuroblastoma model, and these neuroblastoma form metastasis, and we tested metastasis in the kidney. In initial experiments to optimize the model, we used HEC293 cells, but for the final experiments, we also used primary kidney fibroblasts. So here you see our model, which you can call an egg model or something. In the center, you have the tumor, and the surrounding part is the kidney part. And here we even have a 48 well plate, and you see that these models are highly reproducible, so you can test a lot of substances in this cancer model. We tested panobinostat, which is a therapeutic for neuroblastoma. And here you see a live dead stain. In the center, you have the neuroblastoma cells. And on the outer part, you have the kidney cells, the HEC293 cells. And you see that with increasing concentration of panobinostat, the tumor cells in the center are killed, and you have the red fluorescent of dead cells, while the outer part of the kidney cells remain unaffected, so they still have the green fluorescence indicating living cells. We also tested the mode of action of panobinostat. Panobinostat induces apoptosis, so we measured cleaved caspase-3 as a typical marker of apoptosis. And what you can see here, that the central part of our neuroblastoma cells, we have the central part of neuroblastoma cells, and with increasing concentrations of panobinostat, we have an increasing signal of cleaved caspase-3, which means our apoptosis marker, while we have virtually no apoptosis on the outer part. So here is a quantitative evaluation, and we have a substantially and significant higher intensity of apoptosis for the cancer cells as compared to the healthy cells. This is completely different with a different drug. We also used the blasticidine. Blasticidine has a general cytotoxic effect. And here we see that not only the inner part of the neuroblastoma cells, but also the outer part of the kidney cells start to give a signal for apoptosis. And here the quantitative evaluation demonstrates that apoptosis is pretty similar between the neuroblastoma and the kidney cells. So here we obviously have a drug that is not selective for the neuroblastoma cells. So the aim of our model is to develop a platform to test new cytostatic candidates. And here we demonstrate that it's obviously possible to distinguish between substances that specifically inhibit the tumor cells and those substances that also inhibit the non-tumorous cells. In the final experiments, we replaced the HEC293 cell line, which is a cell line again, by primary kidney fibroblasts, and fortunately results were the same. When using panobinostat as a selective anti-cancer drug, we only selectively killed the cancer cells, but we did not kill the kidney fibroblasts. So we confirm that panobinostat can be identified as a substance that is specific for the tumor. So we think that this bioprinted cancer model can be used, first of all, to test the efficiency of a cytostatic drug candidate, and secondly, to test its specificity for the drug for the cancer cells as compared to the healthy surrounding tissue. In our model, we of course have the cancer embedded into a human microenvironment, and this differs from what I explained we usually find in mouse models where we have the tumor implanted into a murine microenvironment. Another interesting finding, which I did not go into depth too much, is that we find substantially different IC50 values for our drugs in 3D as compared to 2D. So usually there are several orders of magnitudes. So the drugs are 100 to 1,000-fold less, have a 100 to 1,000-fold higher IC50 value in 3D models as compared to 2D models. Okay, I will briefly go into another cancer study we did with collaborators that optimize radiotherapy, and they study radiotherapy at a synchrotron microbeam line at the DZ in Hamburg. And their common experiment is that they fix a mouse to the beam, and then they turn on the beam and follow the effect of the beam. So we again printed our cancer model and replaced this mouse with a cancer model and fixed this cancer model in the beam. It was only a proof of concept study. And what you can see here in the live dead stain, again, you see you can really see where the beam goes through the model. So you see the red cells and the lack of green cells in the beam line. And so this is quantitatively evaluated that we have a reduced metabolic activity in the treated cells. And what we also find is induction of gamma H2AX, which is a marker for a repair enzyme that is expressed to repair double-strand breaks that are induced by the radiation. So this is only a pilot study. What we currently do is that we include our printed cancer models into a mouse or into a human lung phantom that reflects the material properties of a human lung. And then we have the cancer in this phantom and want to place this into the microbeam to study the effects of the microbeam on our three-dimensional tumor. Okay, another important point and strength of bioprinting is the high resolution. In biological tissues, of course, we have a vascular system that supplies the cells with blood, with nutrients, with oxygen. And this we can reproduce with our bioprinting approach. Here I show you several models. You have seen this before, and here are two more models where we have a vascular structure in our 3D printed tissue model. So this is a paper which came out only last week, and thank you for the funding. Katrin also already introduced that she could help us with some financial support from her governmental organization. And so we could buy this Lumen X stereolithography printer or DLP printer from this funding. And we produced a vascularized tissue model, which we then connected to a peristaltic pump. So we had a perfusion of our tissue model. So here you see the three-dimensional model, which was designed by the CUT program. We have an inlet and an outlet, and here we have the channel, the tubing that goes through our 3D model. Here you see the model, and here it is connected to the peristaltic pump. We have a reservoir, and we started with a blue ink to see whether the model is perfused. And here you see that with increasing time, we have a nice perfusion of the model with our blue ink. When we then, of course, used common cell media and found that the cell viability increases significantly in the perfused model as compared to the static cultivation of the same model. And then we added, again, a cancer drug, gemcitabine, which can be used. In this case, we have a lung cancer model, so we used a non-small cell lung cancer cell line, and we then added gemcitabine to the model, and there was a substantially higher and statistically significant higher induction of apoptosis in our cells upon perfusion as compared to the static cultivation, and that can be confirmed with caspase-3 as well as with a second apoptosis marker, PARP1. So what we think is that we can nicely introduce a vascular system in our model with a bioprinting approach, and then we can perfuse our model and give a more realistic picture of the biological system. Here, another PhD student shows me his perfused model. So again, here's an inlet, and then here's the tubing and the outlet on the other side. He then produces this model with the extrusion printer. So there's a different production system. So here we have the pneumatic microextrusion. He can then take out the model for this little movie. He didn't work under sterile conditions. And here he has a peristaltic, sorry, that's not a peristaltic pump, but it's rather a micropump. And again, he uses the blue ink to visualize the perfusion of the model. So here you see the model with the vascular structure, and you can introduce the colored liquid and you see that it flows through the model. He casts this into agar, and he has these little micropumps that continuously perfuse the model over days and weeks. And here the center part, this blue part in the center, I should mention, that is where we place a tumor spheroid. So we have the healthy tissue, we have the vascular system with an inlet and an outlet that is perfused with this micropump, and we have a tumor spheroid in the center. And this is where we can now test our cytostatic drug candidates. Oh, for the last few minutes, I'm already close to the end, but give me a few more minutes to introduce our concept of clean bioprinting. As I've already mentioned throughout my presentation, is that we usually talk about, oh, we have an alternative method, we have a new approach methodology, we produce a three-dimensional human organoid, organ model, whatever it is. But in fact, actually, we use a lot of animal components. Most of us culture the cells in FBS media. I already mentioned that many people use Matrigel or Kurreck, that's their brand names, at least some kind of extracellular matrix that is obtained from mice. And we also use gelatin in our models that was obtained from rats or pigs. So I already mentioned Matrigel. I found when I screened the literature last year, I found more than 13,000 studies in the PubMed with the entry with the search term Matrigel, because it's widely used. It's used for all types of 3D cell culture. It's used for spheroid cultures. It's used for all types of stem cell culture. It's used for animal studies. Second big part is, of course, the use of fetal calf serum, and probably all of you know this long discussion on the harvesting of FBS from fetuses of slaughtered cows. And here we suggest alternatives, either human platelet lysate, or even better is a chemical defined synthetic media For matrigel, we can either use human ECM, which is not easily available, or plant material or other non-animal material or synthetic material. And I will give you an example that's not yet published. It's an ongoing project where we try to produce a completely xeno-free bioprinted model. It's supposed to be a liver model, so we use HUH7 cells. We started transferring the cells from the common cell culture medium to a medium that is consists of DMIM and F12, still contains FCS. Then we reduced FCS, and unfortunately, after several splittings, we found that the cells die. So this is already an important message, that if you culture cells under FCS-free conditions for 3 or 4 passages, usually they are still viable and behave nicely. But this is because you have remaining FCS in the cells, even if you culture without FCS. So you have to culture them for a long term, like at least 5 passages, and then you will see that they die, or you have them successfully adapted. And so then my PhD student added several growth factors to the medium, and then he found that they still have a high viability even after a longer period of cultivations. So we had the cells cultured under chemically defined conditions. The next point is that when you passage your cells, you usually use trypsin to detach the cells from the cell culture plate or from the cell culture bottle. And the trypsin commonly is porcine derived, so we use the xeno-free trypsin, and we found that it's very easy to replace. So he determined detaching efficiency and cell viability, and actually there was no difference between the porcine-derived trypsin and the xeno-free trypsin, so obviously it's easy to replace this one. The next is the storage, freezing of the cells. The standard medium is to use 90% FCS and DMSO. This is the standard media for cell freezing. So my PhD student, he tested various compositions and found that in the end, a composition containing PVP has a very high survival rate of the cells after defreezing them again and taking them back into culture. They have nice microscopic properties, and he has a good cell viability. So this is a xeno-free media for cultivation. And finally, we optimized our bioink. We have a collaboration with Johannes Hacketal from Human Touch, and we used placenta in our bioink instead of matrigel. And Ahmed designed a xeno-free bioink, and first of all, he demonstrated nice printability. So here he has printed some of these grid structures, so it's nicely printable. And he also measured the cell viability and metabolic activity and found good metabolic activity in the XCT assay and a high cell viability in these xeno-free bioprinted constructs. So we propose to call these bioprinting approaches without animal components clean bioprinting, which is an analogy to clean meat. And we think that this not only contributes to animal welfare, but also to a human model that only consists of human components, and we don't want to place our human cells into animal microenvironment again. Okay, let me finish by an announcement. Katrin already mentioned that she was so kind to give us some money, which actually is extra work for us, but we are happy to do this work. And in case there are master student, PhD students, or early postdocs from Berlin or around, we're happy to announce a workshop on xeno-free cell culture and bioprinting in early June. And so you can register here in May. What will we have? We'll have a lecture series on different talks on this topic, but we'll also have a hands-on lab course. So this is why we unfortunately only have 10 places in the course. So we will teach you how to adapt cell lines to animal-free conditions, how to do the cell splitting under xeno-free conditions, xeno-free cryopreservation. You saw some examples in my presentation. And we'll also do some bioprinting with xeno-free materials. So if you're around in Berlin, please register, and will be happy to teach you these topics. Okay, with this, I want to summarize. I hope that I convinced you that bioprinting is a very powerful technology to generate objects with a high spatial resolution, and we can produce organ models that we can then use as an alternative to animal testing. We have generated lung models, and I took you through our story of producing different generations of lung models. The most advanced lung models consist of 4 cell types: epithelial cells, endothelial cells, and fibroblasts with macrophage-like cells in between. And we culture them under air-liquid interface conditions, and these models can be used for infection studies. We've also used a liver model, which can be used for cancer studies, and we also currently carry out some toxicity studies. And then I demonstrated to you our cancer models, which we think can be used to differentiate new candidates of anti-cancer drugs with respect to their efficiency and their specificity for the cancer cell as compared to the healthy environment. And the bioprinting also allows to include vascular structure. That is a big advantage compared to other 3D cultivation methods like organoids or spheroids. And finally, I wanted to make you aware of the use of many animal components in our NAMs, and that we should follow routes to replace them. And with this, I want to thank my co-workers again and all our collaborators, and of course also the funding agencies that helped us to carry out our research. And with this, I am thankful for your attention.